Check, check. Okay. Works. Sometimes when I've done it, it takes a while.
Yeah, I mean, whatever you find. Thank you. You weren't on today, right? They just didn't come. Thank you. Again, you can get started whenever you're ready. I think both of these are. I don't know, what do you think about a little low legs or?
This doesn't advance, does it? Or it does? Oh, it does. Okay, good. Oh, that's good.
So you can get started whenever.
Okay. Okay, so I guess we'll get started. Welcome this morning to the talk on glucose and energy metabolism. I'm Dan Donovan. I'm an endocrinologist here, and with a big interest in diabetes, especially type 2 diabetes. And I do clinical trials and various other studies really involved with metabolism and diabetes. So it's a pleasure to be here with you today. Let's see. In terms of disclosures, I do have grant and research support from the NIH and several industry sponsors, Novo Nordisk, Merck, and AstraZeneca. In addition, I've done some consulting and some speaking for Novo Nordisk, Merck, AstraZeneca, and Lilly. If you actually look over my 22-year history on the faculty here, I probably worked in either research or consulting capacity with every company that has ever made or makes or will make an anti-diabetic agent. So you can take that and use it as you wish. So today we have some objectives. We're going to talk about metabolism, define it, the two kinds of metabolism, anabolism and catabolism. We're going to review in a more brief fashion, because you've done this in biochemistry over weeks, the metabolism of carbohydrates, protein, and fat. And I hope that I impart upon you the importance of glucose as the prime metabolic fuel. And we'd like to, after this talk, have a sense of understanding of all the metabolic events occurring during the absorptive and post-absorptive states, sometimes called the fed state and the fasting state. And there's a lot of stuff going on, a lot of interconversions. So I'm trying to keep it as simple as possible because you've done this in detail elsewhere. At least I assume you have. Okay, so just one slide with an overview of energy metabolism. The food we eat is our only source of energy for doing biologic work in our cells. And the foods we eat are carbohydrates, lipids, proteins, nucleic acids. All molecules that are nutrient molecules have energy stored in their bonds. This energy is what runs most biological systems. And the ultimate source of this energy comes from the sun. So plants trap solar energy through photosynthesis, and then plants are eaten by other creatures that are eaten by other creatures, and we eat both plants and other creatures. Just to review, the calorie is the unit of measure of energy in biologic systems, usually with a small c. But when we're talking about human metabolism, we're talking about the big C, a kilocalorie. And it's the amount of energy required to raise the temperature of 1 kilogram of water 1 degree Celsius. And the caloric needs of most organisms are measured in the basal metabolic rate. So, what are the sources of nutrients that we absorb through our digestive system? Well, we've got protein, which has about 4 kilocalories per gram. Carbohydrates also has about 4 kilocalories per gram. Fats have about 9, so more than twice the energy is stored in fats than in the other two. And alcohol does contribute some calories. We don't consider it really to be a nutrient. And so for people following low-carb diets who are not eating any carbs and just using protein and fat, the portions are smaller because they're more calorically dense. So energy for immediate use by cells to conduct their normal metabolic processes, such as muscle contraction, secretions, active transport, and everything else going on. In addition, for the synthesis of structural or functional molecules to repair and replace cells, and also as storage. We can store glycogen in liver and muscle. We can store fat in the adipose tissue. We don't store amino acids. Amino acids are present in muscle, but muscle is not considered to be a storage site for amino acids, although we can call upon muscle to extract those amino acids if we need. So, all living things, from bacteria to humans, conduct metabolism. And it's the ability to acquire and use energy from the environment. Metabolic processes are all chemical reactions that occur in cells, tissues, organs, and organ systems. And there are two main kinds of reactions. Catabolism, which is the breakdown of large molecules into simpler ones, producing energy. And anabolism is to build up, building large molecules from simple molecules, and requires energy input. And this is just a cartoon to remind you of what's actually going on. So, energy balance varies depending which phase we're in, either the absorptive or fed stage or the post-absorptive or fasting state. So energy balance is not continuous. Our energy input is intermittent. So during the absorptive state, it follows eating and 3 to 4 hours after the meal. Nutrients in the bloodstream are plentiful because of all the great things that have been ingested. And the post-absorptive state, or the fasting state, is between meals. And in this case, when there's no new energy input, we have to call upon stores to maintain homeostasis. And so in the post-absorptive state, energy stores need to be mobilized. So this is kind of a summary diagram of what's happening in the absorptive state. In the absorptive state, energy input is greater than output. Glucose is the primary energy source for the cell. So I will repeat things several times. That's not because I'm mentally defective, but it's on purpose. I was taught you need to hear something at least 3 times for it to stick in your head. So when there's repetition, it's like I'm doing the studying for you. So excess nutrients can be taken up and will be stored. Liver and muscle store glycogen. Adipose tissue stores triglycerides. Amino acids can be used to build protein, to build muscle, but not in general considered to be a storage depot. So glucose can have a number of fates. It can be metabolized to CO2 and water. It can be synthesized into long-chain branching complex carbohydrate glycogen. Glucose can also go to the liver where it can be turned into glycerol. Glucose can also be metabolized into fatty acids and can lead to the synthesis of triglycerides. Amino acids can go into muscle, building protein and cells for structural proteins, as well as the liver, where they can be deaminated and converted into fatty acids. And so there's a lot of potential for metabolic conversion. So you've heard about ATP before, but ATP really is the energy currency of a cell. It can be produced from glucose, fat, proteins, and even alcohol, as I mentioned. You've done the calculations many times. I used to hear it was either 36 or 38, depending on what it was, like NAD or FA. It's still there. So that's a structure similar to nucleic acids. You need to convert the ADP into ATP, which is a high energy bond. That removal of the third phosphate releases the energy that we need to do the work in our cells. Blood glucose is more important than fat and proteins in providing energy. It can be used by all tissue cells, and in some of our cells, it's the only option. That includes neurons and erythrocytes. They get energy only from glucose. Lack of glucose we term hypoglycemia. It can lead to weakness, coma, and death. And therefore, it's very important that the liver helps us maintain normal blood glucose levels, or at least adequate blood glucose levels. As I mentioned, There's a lot of metabolic interconversion. Carbohydrates can play a role in many of these reactions. They can be converted to amino acids after going through glycolysis and turning into pyruvic acid. They get transaminated. They can be synthesized into kind of the storage form of glucose, glycogen, in liver and muscle. They can be used to build fats, as I mentioned. And when we need to, we can break down glycogen and use it to make glucose from the liver. And we can also synthesize glucose from other moieties, lactic acid, amino acids, certain of them, the gluconeogenic amino acids, and triglycerides with the glycerol component can all go back to glucose. So, the brain, very important. It is the major glucose consumer. Consuming, depending on estimates, of 120 to 150 grams of glucose per day. It really is essentially the sole fuel for the brain. However, as we'll mention later, during starvation, ketones, and we'll talk more about ketones toward the end, can begin to become a fuel for the brain, but the brain still prefers glucose. There's no stores in the brain. It's not capable of doing the things that the liver and the kidneys are able to do. It cannot use fatty acids as fuel, although fatty acids, after they've been condensed into ketones, can provide fuel. It requires oxygen always to burn its glucose. So not having glucose or not having oxygen leads to the same result: no metabolism in the brain, and the brain dies. It cannot live on anaerobic pathways, and really is one of the most fastidious and voracious of all organs in terms of consuming our glucose. Glucose goes to the brain first, and therefore this supply of oxygen and glucose cannot be interrupted. So you've seen glycolysis again. This is an abbreviated form. This is the first committed step performed by one of the various hexokinases, glucose 6-phosphate. And then the rest of metabolism leading to some reducing equivalents and ATPs, and you're left with 2 pyruvic acids. This beast has a number of metabolic fates. It can go from the cytoplasm to the mitochondria, where it gets converted into a 2-carbon fragment. CO2 is produced. These acyl groups can be turned into acetyl coenzyme A by coenzyme A, and this, of course, is very important in the Krebs cycle. I'm just going to there's a lot of stuff on the slide, but just to make the point, the hexokinase is very important. There are at least 5 different isoforms, and they're different. They catalyze the same reaction, but their pharmacokinetics are quite different. In the liver, hexokinase 4 is called glucokinase. This is also what's happening in the pancreas to sense glucose. It has a low affinity for glucose, as opposed to hexokinase 1 and the other hexokinases. It's not inhibited by G6P. It is subject to reversible inhibition by a protein specific to the liver, so that doesn't happen in the beta cell. And it's transcriptionally regulated by either ATP when it's low or glucose when it's high. And you can see that with the other hexokinases, they saturate very early on. But glucokinase is a great sensor because its affinity is different its activity is different depending on the concentration. And this is just an example of what's happening in a liver cell, similar to what you'll see when I show you the pancreatic beta cell. Glucose gets into the cell through one of the glucose transporters called GLUT, and there's a whole family of them. I have a slide that just summarizes them. I don't really expect you to memorize each of them. But GLUT2 allows glucose into the cell without insulin. So if you've got GLUT2 around, glucose can enter the cell. That's not true of other cells, which require insulin to put their glucose transporters in. And again, it catalyzes the reaction, and it's a nice regulatory system. So those acetyl coenzyme A's go into the Krebs cycle, producing more energy and more reducing equivalents to go to the electron transport chain, making more CO2 along the way. And then this is really important because this is where we get most of our ATPs, and this requires oxygen. So you're not going to be generating ATPs by movement of these NADHs and FADHs down the electron transport chain if you don't have oxygen, and it will stall and you'll get no ATPs. And the end result is water. So that's like the quick review of things that you probably study for several weeks. Just a summary of the process. The production of ATP results in stored potential energy for chemical reactions, but about 40% of the energy is released. And production of heat maintains body temperature and you can actually get up to 60% of energy released, along with the CO2 and H2O. So it's not the most efficient process of energy generation, but we use that heat to maintain normal body temperatures. So we're going to talk about the metabolic profiles of the various tissues, and I already mentioned the brain is the most voracious of our organs, consuming a lot of glucose. It can use ketone bodies in starvation. Glucose enters the brain without the need for insulin. So it's got, GLUT2, GLUT1, GLUT3, depending on which kind of neurons. It's not insulin dependent. Hexokinase is saturated at very low concentrations. And hypoglycemic danger is if you fall significantly below 40. Some people would say 50. And the main metabolic pathways are completely aerobic, including glycolysis, the citric acid cycle, and the electron transport chain. So the metabolic profile in skeletal muscle, depending on how active the muscle is, its preferred fuel changes. So in the resting state, muscles prefer to use fatty acids. In highly active muscles, glucose is the fuel. And remember, there's a lot of stored glycogen in muscle as well, in the fed state. And you can also produce lactate if you're in a more anaerobic, high-use situation, which can then be converted back. And we'll look at one of the slides. So the fuels are used for contraction, active transport, and cellular metabolism. The main metabolic pathways during resting are aerobic, as I just mentioned, and in highly active muscle, it becomes anaerobic, breaking down glycogen and burning glucose. And you can see that in this figure about the various fuels depending upon activity level and what's generated. The metabolic profile of the heart. Actually, the heart prefers fatty acids, but it can use glucose as well. And that fuel is used for its activities, contraction, active transport. It's a totally aerobic metabolism, including fatty acid oxidation and all the other players. The adipose tissue, which we really kind of consider an endocrine organ now, but we're not going to talk about the hormonal aspects of the liver, the fat today. Its major fuels are glucose and fatty acids. It is an organ that requires insulin to get the glucose into the cells. And main metabolic pathways in the fat are all the players: glycolysis, fatty acid oxidation, the citric acid cycle, electron transport. You can also make triglycerides in fat, and you can also break them down as lipolysis. So the adipose tissue really is probably the largest energy store reserve we have, probably 95% of our energy stores. Although it's not the preferred fuel, it can be activated and used for maintaining normal metabolic processes. The kidney. The kidney is very important. I know you've all taken the kidney course, and we know it does a lot of important things, but as a major part of the metabolic system, people kind of forget what the kidney can do. It can do a lot of the things that the liver can do. The major fuels are glucose and fatty acids. That may depend on which region of the kidney you're actually looking at. The fuels are used are through active transport. The kidney can make glucose. People forget that. It becomes very important. In starvation, it can be responsible for 25% of gluconeogenesis. So it's a very important kind of maintainer of homeostasis along with the liver. So under normal conditions, it uses glycolysis, fatty acid oxidation, citric acid cycle, and ETS, all oxygen dependent. During starvation, it can be very useful in forming new glucose molecules from precursors, amino acids that are gluconeogenic, as well as other precursors. So it's a very important organ. This is a busy slide, but it's kind of simple. I just have it there really as like placeholders. In terms of glucose homeostasis in the body, the glucose input is about 250 grams per day. With a dietary intake of about 180 grams, we produce glucose endogenously through gluconeogenesis or glycogenolysis, and the kidneys are filtering plasma, right? So when you're filtering plasma, that ultrafiltrate is going to have the same glucose concentration as the blood. So the kidney can filter a lot of glucose, up to 180 grams per day. Now, what if the kidney just let it go in the urine? Well, at 4 calories per gram, that would be, what, 760 calories going out every day? Well, in our past, when we were hunter-foragers and skinny and just striving to find food, it would be very evolutionary disadvantageous to be pouring out calories in the urine. That's not a good thing. In addition to osmotic diuresis, when you got glucose, water has to accompany it. You can deplete water, electrolytes. That would be bad. So our kidneys have adapted to hold on to glucose. And essentially, in the normal state, the kidneys reabsorb every bit of glucose. Normal people do not find any glucose in the urine. So the glucose uptake again, the brain gets a lot, the rest of the body gets the rest. And just how does it do it? Well, when glucose is filtered in the ultrafiltrate, in the proximal tubule, there's a so-called S1 segment that is responsible for 90% of glucose reabsorption. And then in the S3 segment, about the remaining 10% is taken up. Now, this requires a system of kind of co-transporters. So in the proximal tubule, where 90% is happening, there is a glucose absorption pathway called the sodium glucose transporter 2, and glucose will enter the cell and will depart through GLUT2, one of the glucose receptors portals, and it requires energy, so it's a facilitated transport. So this is considered to be low affinity, but very high capacity. And so it's pretty good at sucking up most of the glucose. And then cleaning up further down the line, still in the proximal tubule, are the SGLT1 receptors, which are also present in the gut. You can reabsorb glucose through facilitated transport as well. And then you shouldn't find any glucose in the urine. Interesting, this is a target of anti-diabetic pharmacology now. We have 3 classes, a class of drugs that blocks this pathway, and 3 drugs in that class. So it causes spilling of about half the glucose in the urine. So people need to drink water. They don't get osmotic diuresis, that's terrible, but they tend to lose weight because they're losing 300 calories of urine in 300 calories of glucose in their urine every day. So it's an interesting system. And there are metabolic mutations where people or families that are glucosuric because they have no functional SGLT2. So it's an interesting target. And just to illustrate, if your reabsorption is at maximum, you will not start spilling glucose until you exceed that. And so in the kidneys, it's about 180 to 200. Normal people really 180. If you change that, such as you have a mutation and your tubular maximum for reabsorption is much lower, say 90, you'll start spilling much earlier. And certainly, as you might hear in diabetes, there's an adaptation of the kidney to hold on to even more glucose, which may be maladaptive. So the kidneys are really very important. So the liver is the big one, though, in terms of integrating everything. It is responsible for maintaining normal glucose homeostasis. It has to make sure that in the fed and fasting states there are appropriate amounts of glucose around. It can do a lot of things. It can synthesize glucose, fatty acids, glycogen, fats, cholesterol, bile salts, protein, urea. It's really kind of the metabolic hub. So carbohydrates are coming in, they're undergoing glycolysis, glycogenesis, the formation of glycogen, lipogenesis, they're going through the citric acid cycle and the electron transport chain for energy generation. And in lower blood glucose states, the liver calls upon its stored glycogen, starts debranching it and breaking off glucose molecules, and it can also synthesize new glucose. Now, only the liver can take glucose that's stored in glycogen and release it into the blood. The muscles do not possess the glucose 6-phosphatase. So muscle, despite the fact it's got about 400 grams of stored glycogen, the liver's got about 100, only the liver's stored glycogen can be produced to be distributed to the rest of the body. The muscles will have to use their own energy stores in place. So in terms of lipids, the liver can oxidize them, generate energy through the pathways we talked about. If there's excess delivery of fatty acids, the liver can condense these into ketone bodies, which we'll talk more about. And when there's too much fuel, then it can be stored as fatty acids into actual triglycerides. So amino acids can be used for protein synthesis. They can be deaminated to form urea. They can be used to synthesize other nitrogenous compounds. And for the gluconeogenic amino acids, the amino acids can become a source to the liver to maintain glucose. Remember, we're always trying to maintain glucose. Just a quick look at some of the metabolic interactions that occur. If the muscle is rapidly metabolizing anaerobically its glycogen to lactate, that lactate can leave and get to the liver. The liver is very good at taking lactate and making it back into glucose, and so you can maintain blood glucose levels. In addition, the same thing can happen with amino acids. If you're actually breaking down amino acids, you can actually re-deliver those products to the liver where glucose can come back up. So there's a lot of metabolic interconversion going on. It's a very integrated system. So let's talk a bit about the multi-hormonal regulation of glucose. So our plasma glucose level is going to be a function of the rate at which glucose appears, and that's either coming through ingested carbohydrates or from hepatic glucose production, and the rate at which that disappears. And most glucose disposal, except for the brain, is happening under insulin. And insulin stimulates glucose uptake into the muscle and into the fat. It actually induces little GLUT4 transporters, those little glucose portals, and we'll see that in a slide. And so you dispose it into the tissue. So anything that affects either the rate of glucose appearance or disappearance is going to affect your plasma glucose level. But it's kind of a complicated story that's getting even more complicated. This is a simplified version, because we keep finding new little mediators that are playing a role. So in response to ingested nutrients, carbohydrates, insulin is secreted. That will help deliver glucose into the muscle and fat. Glucagon, the other hormone made in the pancreas, and we'll talk more about that coming up, is kind of anti-insulin. It's the hormone that's present in the postprandial state, or the fasting state, and it's driving the liver to make glucose. So certainly when there's glucose present, you don't need the liver anymore. So you want to shut the liver off. Turns out the liver does not shut off in type 2 diabetes, and that's part of the problem. And in addition, it turned out, it's only about 20 years ago that this hormone was actually discovered. It is another hormone called amylin, secreted by the same beta cells that make insulin. And it's co-secreted. It turns out to be a neuroendocrine hormone that has a number of effects, including satiety, slowing gastric emptying. And so it's kind of an interesting player. There's even an injected amylin analog. So when I talk about diabetes pharma, I'm not really going to talk about it because it's not used very, very often. But certainly interesting things that could be intervened with. And so it can slow gastric emptying, it can slow food intake. And then in response to ingested nutrients, some other things are happening. Our gut is an endocrine organ. And so we have secretion of what have been termed now incretin hormones, and we're going to talk about what incretins are. GLP-1, which is glucagon-like peptide, because it comes from the glucagon gene, it is nothing like glucagon in its actions. And in fact, GLP-1 gets a lot of attention as a potential, or it's being used as a pharmacologic agent in diabetes actually, well, in various analogs, in that it stimulates insulin secretion from the beta cells. But as opposed to some other drugs that turn on the beta cell, this is physiologic. So it only stimulates insulin secretion when blood glucose levels are rising or are high. When blood glucose levels are normal or low, it does not have an effect. So it's a modulator of insulin secretion, a very physiologically important modulator. Turns out it can also lower glucagon levels. Exactly how it does it is not clear, but GLP-1 will lower glucagon, so it restores that balance when there's food around. And in high enough levels, there are receptors in the brain that lead to satiety. There are receptors on the stomach that slow gastric emptying, so you're not delivering glucose as rapidly. There's another incretin hormone called GIP, which used to be gastrin inhibitory peptide. Now it's renamed glucose-dependent insulinotropic peptide. And it too stimulates insulin secretion from the beta cell, but only the first phase of insulin secretion. GLP-1 does both the first and second phase. That's going to come up again. And so it's kind of a tight system. In addition, there are other neuropeptides being released in the gut that have effects on eating behavior. It's a little too complicated to go through for this purpose of this talk. So, in normal humans, we maintain our blood sugars, even after meals, breakfast, lunch, dinner, at a very narrow range. You never really get higher than 140, otherwise you'd have impaired glucose tolerance. And how is this accomplished? Well, the major driver of glucose metabolism is insulin. And with the meals, there's an appropriate burst of insulin that goes from the beta cells directly to the portal circulation, goes to the liver first before it gets out into the periphery. And so when you're thinking about disease, and you want to keep that in mind, this is what you're trying to mimic. Just a quick mention of amylase again. It's one of the hormones that's co-secreted. Interesting, may have future therapeutic advantages, but right now it's not an easy or very common agent. So this is a schematic of the pancreatic islet cells scattered amongst the exocrine pancreas, the endocrine pancreas in the exocrine pancreas. And you can see there are a number of cells. There are the alpha cells that make glucagon. There are the beta cells that make insulin. There are delta cells. There are gamma cells. Delta cells make somatostatin. A lot of stuff is going on in these islets, but they're tightly packed. And you can see if you stain in this islet for glucagon, it's kind of all over. Insulin in this staining is kind of more peripheral. And what's happening in the beta cell is that insulin is first made as a pre-pro-insulin, cleaved, becomes proinsulin, and then this peptide here, it starts at one chain. There are several disulfide bonds, 3 in total, and this peptide, which is cleaved off, is called C-peptide. It has a very long half-life, so we can sometimes measure it because it's a better integrated measure of insulin secretion. Because once you've actually made insulin, insulin has a pharmacokinetic half-life of about 6 minutes. So once it's released from the granules in the beta cell into the circulation, it's got about 6 minutes to do its work. Once it bonds to its receptor and does all the metabolic machinery, the biologic half-life is probably 40 minutes or more. But the actual presence of that molecule, once it's secreted, 6 minutes, doesn't have a lot of time. So measuring insulin levels is not always a very useful way to get an idea of what your insulin secretion is like. C-peptide is a better integrated measure because it hangs around for a long time. So very similar to what I mentioned in the liver cell, glucose enters through GLUT2, does not require insulin, so it freely enters. In the case of the pancreatic beta cell, we have hexokinase 4 or glucokinase, the first committed step, and it's regulated. And the higher the glucose, the more active it gets. That leads to oxidation, production of ATPs. This channel is very important. When it's closed, when glucose is high, that leads to the calcium channel opening. Influx of calcium leads to the release of insulin, which is stored in granules, preformed granules. This is responsible for the first phase of insulin secretion. Turns out probably the first oral class of drugs for diabetes, the sulfonylureas, and then followed by short-acting drugs, the meglitinides, actually bind to this and keep this channel closed. So their effects on insulin secretion are constitutive. Not a very favored class of drugs, as we'll talk about on Thursday. So with this, we're releasing insulin. And this is demonstrating what I talked about. This is if you give someone an IV, a rat, a mouse, doesn't matter what, you get the same curves. You will see a rapid increase in plasma insulin. That really is essentially the release of the preformed granules. And then, with continued high glucose levels, you then have the mobilization of insulin that's in process in the endoplasmic reticulum, put into secretory vesicles, and you get this second phase, insulin secretion, as long as there's glucose around that needs to be metabolized. So it's a tightly regulated system in normal, and that's why we have this system is so exquisitely sensitive, it can maintain our blood sugars in that normal range without the risk for hypoglycemia or hyperglycemia. So insulin signaling, and this is a really old slide. It's actually much more complicated than this now, but I'm going to try to make it simple. Insulin binds to the receptor. It's got an alpha chain, a beta chain. There's two. It's a heterodimer. When insulin binds to this receptor, it leads to a phosphorylation reaction of tyrosine, which then goes on to lead to further phosphorylation of tyrosine kinase and sets up a whole phosphorylation reaction. Now, insulin is considered to be an anabolic hormone. It builds up. So in addition to its functions in glucose homeostasis, it has other functions in the cell as well. And so it can lead to growth, and we're going to focus today on what happens in terms of glucose. So when insulin binds to its receptor, these glucose transporters are moved to the cell surface in insulin-dependent tissues. Otherwise, glucose really can't get in, except through like mass action. And so this is the GLUT4 transporter. And so these little portals are poked into the cell membrane, and glucose is allowed to come in. And with insulin and glucose transport, we maintain normal glucose, unless you've got type 2 diabetes where that's messed up. And I don't expect you to memorize this, but just to see there's so many of these transporters that are very specific. So the red blood cells have GLUT1, liver, kidney, and pancreas have GLUT2, does not require insulin. This does not require insulin either. Neurons get a few. GLUT4 is really the big player in the insulin-sensitive tissues, and the other ones are there for other functions. So insulin affects tissues differently. In the muscle, the use of glucose and immediate use for exercise or storage as glycogen, exercising muscle can take up some glucose without insulin, although it's probably more mass action. And in fact, the insulin effects last a bit of time. Liver, glucose that's taken up is primarily stored as glycogen, although it could be oxidized if the liver needed energy, but the liver likes fatty acids. It inhibits glycogen phosphorylase. It activates glycogen synthesis. So it prevents breakdown of glycogen, activates synthesis. It inhibits the synthesis of new glucose, because you don't need it when you've got glucose coming in. Otherwise, you'd be having overproduction by the liver. And it promotes excess glucose's conversion into a storage form as fatty acids. In the adipose tissue, which is very sensitive to insulin, it promotes glucose uptake and conversion of glycerol for fat production, and it markedly inhibits lipolysis. When insulin levels fall drastically, the fat cell turns into a lipolytic machine, breaking down all its fat and delivering those fatty acids to the liver. So it just takes a little bit of insulin to control the adipocyte from releasing too much fatty acids. And when there's very low insulin, then the liver, the fat cell, is mobilizing those fat stores. Glucagon, as I mentioned, is the kind of anti-insulin hormone. It too binds to its receptor, in this case a G-protein coupled receptor, leads to a number of signaling reactions. Its major effects are to break down glycogen to make new glucose, and to break down amino acids that are delivered through gluconeogenesis to maintain normal blood glucose. And really, in the post-absorptive state, it's glucagon that's driving the liver to keep providing glucose to maintain a normal level. So it's breaking down glucose, and it's making new carbohydrates. So very important counter-regulatory hormone. So what happens after we ingest a high-carbohydrate meal? Well, you can see blood glucose rise. This is still within the normal range. Rapid increase of insulin, as we might expect. And interestingly, in the normal state, a marked decrease in glucagon, exactly what we want. We got glucose in the gut. We do not need to have the liver make more. That doesn't work in type 2 diabetes. In fact, glucagon goes up, so that's a problem. But this is kind of the normal physiology, and regulated quite nicely. So regulation of insulin secretion and glucagon is kind of complex. Nervous system, parasympathetic stimulates insulin, sympathetic stimulates glucagon. Makes sense, counter-regulatory hormones. GIP, which I mentioned, is one of the incretins. It stimulates first phase. GLP-1 stimulates both first and second phase, as well as inhibiting glucagon secretion. And even cholecystokinin has been demonstrated to have some stimulatory effect on insulin secretion, although not really listed as an incretin anywhere I've ever seen it. So I mentioned incretins a lot. And just to give you some history, because it's really quite elegant, it came from the fact that if you give an oral glucose load and then you measure insulin secretion, look, we're using C-peptide, smooths things out, you get a brisk increase in insulin secretion. What if you give the same amount of glucose so that that glucose gives you the same plasma curve? So the beta cells are seeing the same. So this time we give intravenous glucose. We do a really good job, or Dr. Knott did a really good job, this is classic, and did a really good job demonstrating that yes, there's insulin secretion, but it's blunted. So this difference between the insulin secreted for orally ingested nutrients versus injected nutrients was termed the incretin effect. And lo and behold, over several years, it was discovered that GIP and GLP-1 are the two main incretins. There's probably others. They account for about 80% of the incretin effect we see after ingestion of nutrients. GIP comes from the proximal small intestine. GLP-1 comes from the distal ileum and colon, but it's secreted rapidly after ingestion of a meal. So there must be some neurosignaling that's happening in the gut to lead to these incretins, which go up very quickly. Now, both of these agents have about 90 seconds to do their thing, which is stimulating insulin secretion in a glucose-dependent fashion. So it won't stimulate glucose if the glucose levels are low. So they don't have a long time to do their thing. So actually one of the drugs that I'll mention actually blocks the breakdown of these incretins, so they last a little bit longer and can stimulate insulin secretion for longer. GLP-1 gets a lot of attention because in addition to its effect on appropriate insulin secretion in a glucose-dependent fashion, it also lowers glucagon, and it thereby, with higher insulin, low glucagon, that's a signal to the liver, stop making glucose. It slows gastric emptying. There are receptors for GLP-1, and there are central receptors that, with high enough levels of GLP-1, promote satiety and reduce appetite. So this lists major functions of the hormones in the metabolic pathways. Insulin, as we mentioned, promotes storage and growth. Glucagon mobilizes fuel. Epinephrine can mobilize fuel. It can stimulate glycogenolysis and fatty release. These are all glucagon, epinephrine, cortisol considered counter-regulatory hormones. So if you drop blood glucose rapidly, you'll see a surge in glucagon, epinephrine, and cortisol, which has a longer-term effect. You know, it's a nuclear receptor. It takes hours to have an effect, but it will also lead to amino acid mobilization and gluconeogenesis. And thyroid hormone is also important because it regulates our basal metabolic rate and our energy expenditure. Okay. And just a quick mention, one of the molecules that's considered to be an integrator of metabolism, this is called the AMP-dependent protein kinase. And when AMP levels are high, there's not a lot of high phosphate bonds, right? So it's a low energy state. It shuts off synthesis and all the energy in terms of stimulation. This is present in many different cell types. In the liver, we talk about it a bit because I'm going to say that's where the drug metformin actually seems to work. But it's kind of a regulator of metabolism. And I'm not going to talk about it anymore, just so if you ever hear the name, it's also the target by which we believe that metformin works. So after we've eaten and our energy import is no longer prolific, we have to call on the systems we just talked about for mobilizing glucose from glycogen in the liver and using fatty acids and amino acids from other sources. So stored nutrients are broken down and mobilized. Insulin is the hormone of the absorptive state. Glucagon is the hormone of the post-absorptive state or the fasting state. And epinephrine and sympathetic nervous system activation also can play a role. And there's actually a differential regulation, which I'm not going to spend a lot of time on, in the liver, that glucagon will stimulate the breakdown of glycogen. Because the liver does have glucose 6-phosphatase, it can be released as glucose. The muscle does not respond to glucagon. There are no glucagon receptors. It can respond to epinephrine, and it will break down glycogen to glucose 6-phosphate, which can enter the metabolic pathways and generate energy locally. Pyruvate, it could go to lactate too if it was like anaerobic metabolism. And this is just a little idea of looking at liver glycogen through the day. In general, we have about 100 grams stored, but depending on how long it's been between meals, that can be mobilized. And after overnight, and especially in a couple of days, you've probably used up most of your glycogen. And then you have to turn to other sources of maintaining glucose levels. So, lack of insulin in the muscle leads to protein breakdown. Remember, insulin is anabolic. It's anabolic to muscle. It's anabolic to fat. When it's not there, catabolism starts. Amino acids are not stored for energy, but we will ravage our muscle our lean body mass in order to maintain glucose levels for the brain. This is not a healthy thing to do. It's useful in the short term. And again, it generates a lot of stuff, including reducing equivalents for electron transport chain, ammonia, which may not be so great, and has to be converted to urea. And also, these can be turned into keto acids and metabolized in the citric acid cycle. In the fat, remember I said fat is exquisitely sensitive to insulin. When insulin levels drop, and if they're profoundly low, then the fat cell goes wild, sucking up all its fats, releasing it as free fatty acids and glycerol. And this excess fatty acid goes to the liver, and it begins to be metabolized. When it gets down to acetyl-coenzyme A, it can form ketone bodies. I have a diagram showing what's happening. And these can actually be sent out and can be alternate fuel for certain tissues. Glycerol can be metabolized because glucose becomes glycerol. But this is the kind of breakdown state. You can see what's happening. Glucose production by the liver and kidney during the first 10 days of fasting. See, in the beginning, most of it's from glycogen. And then we're getting fatty acids and glycerol over time to a more steady state. The kidney amps up its production from about 25% of gluconeogenesis to approaching 50% from mostly amino acids. So the kidneys are very important in maintaining normal glucose, especially in the fasting state. So if you look at what happens in starvation over a 6-week period, you're really burning a lot of the fat stores, 90% of the calories. You can generate these ketone bodies, which are acetoacetate and beta-hydroxybutyrate, which are very strong acids. And they can provide a fuel source. And the brain can begin to use ketones early on in starvation and can use either acetoacetate or beta-hydroxybutyrate. Only acetoacetate and acetone, which is a ketone but not an acid, can be measured by the usual reaction, the nitroprusside reaction, where you do a dip or put it on a tablet and it turns purple. Beta-hydroxybutyrate is not measured. You can now get it measured. So if you're really reduced, you've made a lot of beta-hydroxybutyrate, you might not be able to find it, but you'll have an anion gap if there's a lot of them because you have an unmeasured anion. And this is what we're talking about. We're condensing acetyl-CoAs. We're having an acetoacetyl-CoA. That can be further added to. All these little CO2s that are bound to coenzyme A can become both beta-hydroxymethylbutyryl-CoA, becoming the first of the keto acids, acetoacetate, which can also break down into acetone, which you might be familiar with the smell of nail polish remover, or if it's on someone's breath, if they're in diabetic ketoacidosis or they're doing the Atkins diet successfully, acetone is released, but it doesn't, it's not affecting pH balance in the blood. And beta-hydroxybutyrate, even in a further reduced state, is also present. And if things are really being driven, you can have mostly beta-hydroxybutyrate. Now, when you're making tons of these, it's not good. A little bit, not so bad. When you drop your pH, it's not physiologic, and there's respiratory compensation. People who are ketosis, especially diabetic ketoacidosis, are breathing very rapidly, trying to blow off CO2 and maintain pH. And this is not a condition, in the complete lack of insulin, it could lead to death. So again, just a summary of the fuels that, I didn't realize it was such a build. Glucose utilization by the brain begins to drop. This is the 40th day. Ketone bodies become a big source. Adipose tissue is breaking down. If you kept degrading protein, you'd be losing all your muscle mass, so that begins to slow down. The liver's making less glucose, but it's making a lot of ketone bodies, and the kidney ramps it up. So there's a lot of adaptations that are occurring in starvation. And this is just kind of a summary of the things we've just really talked about for you guys to take a peek at. So ketone bodies are overproduced in both diabetes that is uncontrolled, where there's really low insulin levels, and you're mobilizing all these free fatty acids from the adipocyte stores. They're broken down, they're generated, the glycerol is being used for glucose, the free fatty acids are being condensed, and you end up with a lot. Glucose, these ketone bodies, these keto acids, are essentially a soluble form of lipid energy, whereas lipids are not soluble. So they can get into the brain can use it, it prefers glucose. When this goes too far, you can get very sick. And just again, a summary of what's happening in the post-absorption state. You've got these stored macromolecules. You can break down protein from amino acids and do all the things that amino acids do. Adipocyte tissue, you break down your triglycerides, you're making fatty acids. They can go to the liver and become ketone bodies. Glycerol can actually go back and become glucose, or it can be metabolized further. The liver's breaking down its glycogen and synthesizing new glucose if it needs to. And again, the glycogen in muscle can only be used in the muscle. It cannot get back out to raise blood glucose concentrations. So this is just kind of the last slide showing the integration of all these metabolic processes that are actually happening to maintain homeostasis. And in the normal state, it's done quite well. But you're going to be hearing about diseases that mess this up. So I'd like to thank you for your kind attention. I'll take questions if things are unclear. And have a good day. See you on Thursday, I guess. Is there a question? Thanks. Hi. Acetoacetate and beta-hydroxybutyrate are strong acids, so they dissociate into acetoacetate and hydrogen ions. If you pour hydrogen ions into your circulation, you're going to drop your pH. So it's like the Henderson-Hasselbalch equation. You can do calculations on it, but it's really they're dissociating. It's like pouring an acid into your blood. You're welcome.
Yeah.
Hey there, how you doing? Good, good, good. Sure. Yeah. Well, I think, in those 6 minutes, it gets to bind to receptors, and once that process is started, so the biologic effect of insulin will last much longer. Which is why if your pump malfunctions or your tube gets clogged, you don't go into decay in a second. Or you can go into decay more rapidly than injected, because your only insulin is the one that's going through it. It's so interesting that these molecules that have such short half-lives can be so important. Uh-huh. Yeah. And that's one of the problems too. The way we deliver insulin is subcutaneously. And I mentioned that insulin is secreted pulsatilely into the portal circulation, goes to the liver first, where some of it's actually extracted before it comes back out. So we're always probably hyperinsulinemic. We're not mimicking normal physiology. Insulin secretion is in pulses. But we're doing a lot better with glucose. Do you use the sensor? The newer ones, like the Dexcom G4, are really amazing. And people are actually working on closed loop systems where your sensor talks to your pump, and you don't even have to be involved. So, yeah, some exciting stuff is happening.
I'm sorry I didn't get to the slides.
No, it's okay. We should.
No, no worries.
One in Mass General, there's the artificial pancreas project that's happening
They got them,
through
so.
West Virginia, and actually at Sinai, one of my colleagues at Sinai is working on that one. Because I think the bionic pancreas is also infusing glucose.
No, I know they're not exactly the same.
Is it putting glucagon in as well?
Some of these I'm going to click right through.
The artificial pancreas program is not.
I'm going to just skip right over them. They don't need to
Yeah.
go over group 4 and, just
So I think, things are very exciting in diabetes, both for type 2 and type 1. I think we're going to see some amazing things happening that'll make your life better. I take care of a lot of type 1s as well as type 2s. And definitely would like to make your lives easier.
Yeah, because I've seen some of the classes are available,
sure.
I think. But you're right, it was not so easy to navigate that system, but I'm sure I could figure it out.
Yeah, right now, I think a lot of the type 1 studies at the very center are looking at kind of immune tolerance in the recent diagnosis period. So Sinai is the only place that's doing artificial pancreas stuff.
It's a lot of paperwork, right? I mean,
Carol
it's
Levy's
hard.
great.
Yeah.
She was a Joslin fellow when I was at the Brigham, but we rotated through Joslin, and Joslin ran our diabetes center at the Brigham. So she's like terrific. But you can always email me. I mean, I'm fine. DSD.
Oh, there is.
All right, great. Thank you.
Take
Take
care.
care. Do you have a question too? Sure.
So we have only 1 or 2 a year, and so that's 3 to 6 at our max. So, like most years, recently we've had 2, but then there'll be a year where there's only 1. But this year there's only 1. It depends on the funding and I guess what they want to do. So, yeah. So. Who's the program director? It's still Sharon Overfield. So she's the division director and program director, which is impossible, you know. So. Yeah, and they only give you 10%, at least in B. You're given 10% to be the program director.
Making myself.
No, our salary is definitely lower than you guys, so for sure they're just underestimating the amount of time they're willing to give us. I'm listening. Oh, nice.
But it depends on, I think there's a max.
Oh, sure.
25%
Real
to
salary.
a party averages.
Sure. That makes sense. I can just, can I just plug this in?
Yeah, it's right there.
Ah, perfect.
They're not used one type one. So they're really only. They're kind of adequate.
Okay.
So they're all recorded and everyone listens to them.
I know. I just realized that as you said it, and I thought, oh my goodness. All right, let's see.
Do you want me to? I think it's under
Your
the
computer.
Yeah.
Did you post that pharmacology syllabus?
No, and I thought you.
Yeah, I'm just adding the Tansium and Trulicity. So it's good. Just post it on my show. I'll just mention it. So they're not responsible for it. But it goes into more detail. Sure. More detail. You know, when you're actually treating somebody, you always take certain issues. But until you get very and I definitely have it. I just, I gotta send you my slides. Huh?
Wow. Where do you save it? Is it in the how do you in the cloud? Where is it?
No, it's there. But I finally asked them for more.
And they'll just give it to you?
Well, I had to ask. I'd be like, I have things on here I cannot delete. So they were like, fine. But I've been up until last week, I was just deleting
It's
big
so
files.
yeah. No, it's so hard. I need to stand on something. I think. Feels silly, but
There used to be a bench here.
I feel like I definitely can't see over this computer to see people.
Is this enough? Or? I don't think so. I don't know what that is.
Yeah, I'll be fine. I guess they'll just have to. They don't have to start yet. Can I go?
Yeah.
Okay.
Thanks so much.
Sure. Good morning. Okay, I think we're going to get started. If you guys just want to have a seat. So I'm Mary Pat Gallagher. I am a pediatric endocrinologist, and I work at the Naomi Berry Diabetes Center. And so Dr. Donovan spoke to you a lot about the functioning of different hormones in maintaining glucose homeostasis and energy balance. And so what I'm going to talk to you about today is to sort of introduce the clinical implications of that a little bit more, and specifically with more attention to type 1 diabetes. And then you're going to get a fantastic lecture about type 2 diabetes from Dr. Leibel. And I don't know, I think that might be, it's in your next class, I believe. So right after this class, you get introduced then in more detail to type 2. And type 2 is what you guys are going to mostly see. 95% of the people in the United States who have diabetes have type 2 diabetes. But what's going to make you, even better than the excellent physicians you'll be working with is that what we want you to know is that although 95% of the people you may encounter have type 2, 5% won't. And so you want to be able to identify who those people are, understand the differences in the pathophysiology behind why they have high glucose, and know what needs to be done differently so you take good care of the patients. And so some of the slides are a review of what Dr. Donovan did, and so we may skip over those, and you can interrupt me at any time if you have any questions. I have no conflict. Nobody is to any of this, so nothing to disclose. The objectives for this particular lecture are that I'd like you to be able to know exactly what are the diagnostic criteria for diabetes. They are not different between different types of diabetes. When you diagnose diabetes, there's one set of diagnostic criteria. It applies to everybody. So I'd like you to know those. I'd like you to be able to discuss at least generally the differences between the pathophysiology of type 1 diabetes and type 2. And most importantly, while recognizing that in terms of your clinical interactions, understand that insulin can never be eliminated from the regimen of a person with type 1 diabetes, okay? For some of the reasons that Dr. Donovan went over about how critical insulin is, not just to blood glucose control, but to energy homeostasis. Everybody needs insulin. And you'll see more why when you have an absolute insulin deficiency in type 1 diabetes, that means exogenous insulin can never be removed. People with type 2 diabetes, you might be removing their exogenous insulin when they're fasting for a procedure, but that's only okay because they still have some. Everybody needs insulin. And then to be able to understand why we try in type 1 diabetes to maintain such tight control and what the benefits are of that. Okay. So diabetes in general, as I said, is, regardless of the etiology, what all the different types of diabetes share is the one thing, which is high blood glucose. And it's sort of a balance. No matter what you're talking about, it's always going to be how much can you make versus how much do you need. And so no matter what, it would seem that the amount of insulin that you're making is not enough. Okay? All endocrine hormones are going to work that way. You can only say what a normal hormone level is in the context of whatever the end organ is it's supposed to be helping, right? So I don't know exactly which endocrine lectures you've had so far, but calcium and parathyroid hormone, you can't say that the parathyroid hormone is low or high unless you know what the calcium is, because it should be something different depending on what someone has. If your calcium's too high, your parathyroid hormone should be lowering itself. And if it's low, the parathyroid hormone should be increasing. Well, insulin and glucose work just like that also. If my glucose is 67, which is fine, but trending down, my insulin should be off. I shouldn't measure any. Meanwhile, if my blood sugar is above normal, no matter what, that was an insufficient amount of insulin. It could measure 1,000, but it's obviously not enough, or my blood sugar wouldn't be elevated. Okay? So no matter what, if you have a high glucose, the insulin level is going to be determined to be insufficient. Now, in type 1 diabetes, which we'll go over in more detail, it could be undetectable. You don't make any. You have an absolute insulin deficiency. In type 2 diabetes or other types of diabetes where insulin resistance is playing a role, you could have a level of 300. Your blood sugar could be 280 and your insulin could be 300. Now, even though it's not in the normal range, it's still insufficient for that person or their blood sugar wouldn't be above 200. So it's kind of a resistance versus how much you can produce. And there's always going to be a combination. We try to neatly divide it, type 1, type 2, but the truth is both of these factors are going to be important in everybody. If you were to try to identify in a mixed kind of picture, which you will see a lot, what makes something type 1? For sure, if you have markers of autoimmunity, that's type 1. There's a little bit of argument about that, but because 2% of the general population has a positive antibody called GAD, and we'll go over that, but it's a super common, non-specific antibody to a neuronal antigen, and so it doesn't absolutely mean someone is going to have beta cell abnormalities, but so it's not 100% clear, but most experts at this time would say if you have a marker, if you have diabetes and you have an autoimmune marker, that's type 1. So even if you have relatively high levels of insulin, because you have insulin resistance also, they would still call it type 1. And then we touched on this a little bit, and we'll talk more about the fact that type 2 is associated with the resistance to insulin, but there's no known autoimmune destruction. Okay, so criteria. The criteria are pretty straightforward. If your blood sugar in the fasting state is 126, that's consistent with diabetes. You'll get a lot of people who are concerned because they'll get a blood sugar of 140 and it'll say H next to it, and you'll get a lot of calls, this person has a high blood sugar. But when you take the history, the patient's eaten. 140 is not high if you've eaten, okay? So 126 is only high if you haven't eaten in 10 hours, so you're fasting. So that's one criteria. A second criteria would be if you did a glucose tolerance test where you would give an adult, it's adjusted for weight in children, but for an adult, you would give 75 grams of a liquid glucose load, and then you would see what happened at 2 hours. You could do a lot of other little measurements in between, and we do them for research studies all the time to see what your insulin production levels look like, but we do not clinically, we don't use them for any diagnosis, okay? So it's the 2-hour blood glucose level after a fixed glucose load that will give you a diagnosis of diabetes if it is 200 or more, okay? So those are two possible ways. And the other is if you just happen to get a lab and the blood glucose, they call it a casual glucose level, is over 200. That also is going to be significant if, and only if, the patient has symptoms, and it's kind of hidden down there, so you might just want to circle it. The only way that that's good enough to give you the diagnosis is if they also have symptoms, not if they just happen to come in for blood work because, you know, they broke their arm and they need surgery and you see the blood sugar is 210, but they have no symptoms, then that doesn't meet the criteria. But if they came in and said, I can't sleep at night, I'm getting up every 2 hours to urinate, and I've lost weight, and I'm so hungry, and I'm eating all the time, and I can't seem to gain any weight, and then they have a blood sugar of 300, that's enough, okay? So those are the blood glucose levels that would give you the diagnosis. And then in 2011, which is getting longer and longer ago, and I guess it doesn't matter, I used to refer to it, we just changed, but that's not really just changed anymore. The ADA included an A1C as a screening that you could use it to diagnose diabetes. We didn't used to do that. we said it really matters more what your glucose metabolism is, and we didn't use A1C, but now you can. So if you have an A1C of 6.5%, you don't need any of these other things. So each of these individually, by themselves, is enough. You don't need all of them, any one of them. Strictly speaking, there are a few restrictions to this information. One is you can't be sick, right? So if someone's in an extremely stressed state, they just had a car accident, they just broke their arm, they are on steroids, like that doesn't count. You can't diagnose someone with diabetes in that period of time, which makes sense. So there's something else going on. And the other is if the person is asymptomatic, so not this last one, and you have an A1C elevation or any of the blood glucose levels we talked about, but no symptoms, you need on two separate days. You need to have these twice to prove it wasn't a fluke. So the person has no symptoms but has a blood sugar of 201 on a glucose tolerance test. If you really want to diagnose them by ADA criteria, you have to do one of these other things on a different day. It could be the A1C, it could be another, a fasting plasma glucose on a different day. You wouldn't have to do another glucose tolerance test as long as it met the criteria here, okay? I don't know if you guys will get here in this lesson, in this group of lectures, anything about pregnancy and blood sugar levels, but they're actually very different. They have many different, stricter guidelines about what's normal in pregnancy and what's not. So we're not going to talk about that here. We're going to talk about people who are in the non-pregnant state, but so that would be separate. This applies to all people who are not pregnant. Okay. So the common types of diabetes, as I said, type 2 diabetes is the most common in the United States. It's almost everyone you've probably come across probably has had type 2 diabetes. Type 1 diabetes is a much less common form. It's pretty much an orphan disease, honestly, and it is becoming more prevalent, though. Like all autoimmune diseases, we're seeing a little bit of an uptake and surge in that, and it's not clear why that is, but it's not going to come close ever to the number of people with type 2 diabetes. Now, in type 1 diabetes, as we said, you have the beta cell destruction and autoimmunity and an absolute insulin deficiency. In type 2, in addition to that insulin-resistant state that causes you to need so much more insulin to keep your blood sugar normal, you also have a progressive insulin secretory defect. So if someone else might be able to have the same level of insulin resistance as you do, and they can make their insulin level go up to 1,000, and their blood sugar never goes over 140 with the glucose tolerance test. It's not normal to need a blood insulin level of 1,000, but they don't have diabetes. They just have severe insulin resistance. But why the next person at the same increased weight, let's say, same insulin resistant state genetically for all the different modifiers of insulin sensitivity, we know there's lots, that person maybe used to be able to make 1,000 units a day, or I'm making that up, but in terms of percentages, but then slowly, then 800, 600, 400, 200. So even though they're hyperinsulinemic by the time they're diabetic, they are also acquiring an insulin secretory defect over time. And after 20 or 30 years of even having diabetes, they'll continue to decline, and ultimately you stop making insulin, even at the end, as an end stage. So even people with type 2 diabetes, they'll become insulin deficient, and they'll need insulin. They didn't develop type 1. They have now insulin deficient type 2. Because when they get replaced with insulin, their doses are going to be enormous. They're still just as resistant as they were before. Okay? So we'll talk a little more about that. And then there are the less common things. Other specific types of diabetes, we're learning more and more about different monogenic forms of diabetes. There's a term that's a little bit outdated, but you still might see it. It's called MODY, maturity onset diabetes of the young. We don't really use that term anymore because the way that it was coined was they saw that there was a group of adult diabetes patients with type 2 who seemed to not behave like the typical type 2s. They didn't look exactly like them in terms of their progression, and they identified that within that group, they became insulin deficient more quickly, and it wasn't autoimmune, and they found these genetic defects. So maturity onset of diabetes of the youth. They used to call type 1 juvenile and type 2 adult onset. Now we know they occur any age. We'll look a little bit about epidemiology of where you see it most commonly, but you can get either, and we don't use those terms either anymore for this reason. But they thought, wow, this is juvenile diabetes in these adults who looked like they had type 2, so they called it maturity onset diabetes of the young. But now we realize there are kids with pictures that look a lot like type 1 with insulin deficiency, who in fact have monogenic forms of diabetes. There are adults who look a lot like type 2, who in fact have monogenic forms of diabetes, and we keep finding more and more of those. Some of the genetic defects are in beta cell secretion, their ability to make more insulin, and some of them are in sensitivity. They affect sensitivity, and you get insulin-resistant states. Other things, a lot of people that we'll be seeing in the hospital especially have had a lot of medications. And a lot of the medications we give for different disorders, particularly chemotherapy, can be pretty toxic to the beta cell. And you can become insulin deficient because of those. Also, any diseases of the exocrine pancreas, like you would see in cystic fibrosis, for instance. It's not exactly clear why, but people who have cystic fibrosis, eventually their islets get destroyed. If it's local inflammation, if it has some genetic marker, we're not really sure. It's an area of intensive study, but they develop insulin deficiency and diabetes, usually after the second decade of life. And then I was thinking about it when Dr. Donovan was talking to you about the counter-regulatory hormones. It's all about energy and balance. So insulin is something when you have plenty of energy coming in, lots of glucose, there's a lot of it, your insulin levels are almost going to direct where to go. You're going to go to the fat cells, the muscle cells, this is going to go to the growing child is making bone, the rest of it's being stored as glycogen, and so fat. Whereas all the counter-regulatory hormones, if you haven't had any intake of any energy, you're fasting. Every night we fast for 12 hours. And I'll tell kids with type 1 all the time because they think, but I didn't eat, so I didn't need any insulin. But you always need insulin. Your body is always in need of energy. And so your body, when you are fasting 8 hours at night, it's constantly releasing the glucose from someplace. It's very smart. Your body's stored all this glucose in your liver, and it's constantly going to be releasing it for you. And at the same time, your beta cells would be constantly releasing a little bit of insulin all the time so that the glucose could be utilized by the body's cells. So I tell them that no matter what, to stay alive, our bodies always need glucose and insulin together in sufficient amounts to at least meet your basic metabolic needs. Okay, if not process a cheeseburger. Yes, you don't need a big shot, right? But you need something. So in that context, you have a lot of other hormones. You've got the glucagon, you've got catecholamines, you have cortisol. So any diseases of any of those hormones with excess production of those hormones are all possible causes of secondary diabetes. So if something looks really a little bit off, something is not right when you're seeing somebody with hyperglycemia, then you would look, does this person have any markers of Cushing's? Is this a secondary mild hyperglycemia? Oh yes, they're hypertensive and suddenly amenorrheic and has acne. Like those are things that should point you in the direction of this is not the typical, adult patient who's overweight with type 2. Most of the time, they're just going to be type 2 patients, but you should be thinking about the other potential things that happen less commonly, and if there's anything that should point you in that direction. A glucagonoma, same thing. It's normally designed to increase your glucose levels, but if it's in excess being produced, it's going to raise your glucose levels inappropriately. Okay? And then gestational diabetes mellitus is officially diagnosed when you have never before had the diagnosis of hyperglycemia, and for the very first time, the glucose is elevated during pregnancy. Now, that doesn't mean that it really is only going to be seen in pregnancy, and that's why when they're after they deliver, they're followed. Because you could easily present with your type 1 diabetes initially in pregnancy because someone screened you, or type 2, and it doesn't really go away. We're going to skip over this because you did this with Dr. Donovan, and we'll just skip to this. So this is a profile, or a profile, of an average person's insulin secretion if they were eating 3 meals a day, right? So you can see that they have some what we call basal or background insulin overnight. It's not that you don't need any, you definitely do. You need background insulin. And then when you eat, your body has to make an additional sort of surge of insulin and try to match the insulin to the food absorption. So as your body is absorbing all of that, glucose and protein and fat, you are going to have to release insulin at a similar rate. And because of all of the beta cells are remarkable cell. And what Dr. Donovan was telling you about the GLUT receptors, the hardest thing to replicate is the sensing function of the beta cell. You can make anything make insulin and release it. That's no problem. People have been able to do that for many years. You can make the liver cells do it. Any cell could be given that machinery, insert the gene, let it release it. But too much insulin is worse than none. We take people's pancreas out if you make too much insulin. We'd rather you have no pancreas than make too much. So the real magic there is that sensing of when I need to release it and when I need to stop so that I don't become hypoglycemic. And you can see it's a marked drop. And then you increase the next time you eat, and then again down. Now these little blips, those have to do with if you are absorbing your food a little more slowly. And that happens when people eat more protein and more fat. It also has to do with some of the other hormones that Dr. Donovan spoke to you about in terms of absorption of food. So if you have decreased, if you have a pure carbohydrate meal, or a glucose tolerance test is basically liquid sugar. That is the most rapidly absorbed type of carbohydrate. But if you have pizza, they've done studies, people digest and absorb all the calories from pizza over 8 hours. Okay? So your beta cells can handle that. It just stays on watch. A little more is coming, I'm going to put out a little more. It stopped, I'm going to eliminate it. And while your body is in charge of making insulin, within 6 minutes, if your beta cells stop releasing it, your body has metabolized it, and it's gone. 6 minutes. For someone who's taking exogenous insulin, they're putting it in the subcutaneous space, right, in the fat layer. And even the rapid-acting insulins that we have, at least 3 hours you have to live with that decision. Whatever we administer, it is there for at least 3 hours. Okay? So we tried to develop insulins that look like this, but the most rapid ones, still you have to deal with it for 3 hours. And you can see, probably that's not needed, but that's as rapid as we have right now. And so some of the glucose sensing studies that have been used with artificial pancreas studies, right? They're looking at continuous replacement of insulin in as physiological a way as we can do it with an insulin pump and using different sensors and adjusting the insulin administration. And we still can't quite get this right. There are groups that actually inject the insulin intraperitoneally. And I thought, oh, that's going to be close. You're almost giving it into the bloodstream. That's got to be close. No. I mean, it's better, but it's not like this. We can't control the glucose levels the way that the body could if it could do it itself. Okay. So this is just a little schematic that's really showing that if you could make the insulin that you were supposed to, you would be doing two things that would keep your glucose levels and your body in energy balance. You would be controlling the release of glucose from your liver primarily by inhibiting glycogen breakdown, okay? Because the insulin's being secreted because you have plenty of glucose in the blood. It's saying, we don't need you to break down any glycogen for us. We're perfectly fine. And then as the glucose levels drop and your insulin level drops, That's a signal to the liver that maybe I should start releasing some glucose, break down some glycogen. And it does, and you can use that for energy. And in terms of muscle and fat, it can then also use glucose. And you wind up with normal glucose levels and no glucose in the urine. So in type 1, or honestly in anything where you have an insufficient amount of insulin for your requirement to keep your blood sugar normal, if you have insulin resistance at the level of the liver, then it doesn't know to stop releasing glucose. It thinks, oh, I don't see any insulin telling me you have enough, so I'm going to keep releasing it. So you keep getting glucose release. And now you have hyperglycemia and less energy production, right? Because you can't get the insulin's not there as far as the body is concerned because it's so resistant to it. So then you're not able to get that glucose into the peripheral tissues either. And so I tell people when they first get diagnosed, yes, the symptoms that are bothering you, that you're up all night urinating, that you lost weight, you're exhausted, the high blood sugar is in part responsible for those, partially because when glucose exceeds the renal threshold, right, it's going to pull through osmosis all this water out with it, and that's what causes the increased urinary symptoms. And then, as a consequence, your brain is intact, your thirst mechanism, it kicks in and tells you drink, drink, drink as a secondary, right? That's appropriate. So now you have increased urination and increased thirst, and what's worst, which they're unaware of, is they have no energy. And they're literally breaking down their body, they're losing fat mass, and they're starving, they're catabolic, which Dan had mentioned in his lecture. So that's really the worry. If you have a blood sugar of 400 and an elevated hemoglobin A1C, which is the average, a reflection of your average blood sugar level over 12 weeks, for years that's going to put you at risk for secondary complications, right? And you're going to get a whole lecture on those. But what's really concerning right here and now is that you need insulin and glucose to get energy and stay alive. So if you really don't have enough, it's not the glucose level that's dangerous, it's this metabolic state of starvation. Okay. In type 2, you have the same sort of symptoms, perhaps, because you have the hyperglycemia, and you have less glucose entering the peripheral tissues, so you might have some weight loss, and you have excessive release from the liver as well because of the insulin resistance at the level of the liver, but you don't have quite as much I would say disarray in terms of your basic needs. We'll talk a little bit about decay in a minute. You certainly can see in type 2 diabetes, but you don't see as often. So these are the clinical manifestations: polyuria, polydipsia, polyphagia. And that's all because of the insufficient insulin. Your body, they call it starving in the midst of plenty, starvation in the midst of plenty, right? Your blood sugar is 400, there's plenty of glucose out there. So why are you losing weight and starving? Why are you cutting, because you can't use it, because you have no insulin. So when you're trying to think about the typical presentation, and of course nobody's really typical, but things that would make you think more one type versus another, type 1 is more often going to present when someone is acutely ill. So they come in, they look sick. They are usually not overweight, although they can be. They're a subsection of the general population, and as the general population has an increasing BMI, so do people with type 1. They're not different in that way, but typically you'll see that you would expect them not to have insulin resistance markers, one of which is weight, right, excess weight. Another one would be acanthosis, right, where you have that thick, sort of dark, velvety pigmentation in flexural areas. You can see it in the neck and the axillae most commonly, but people have it in their fingers, perioral. You can have quite marked signs of insulin resistance. You would not expect that in a child with type 1 diabetes because typically resistance is not part of the pathophysiology of type 1. It's a deficiency. Then there are little peaks. Typically, if you're prepubertal, you don't have type 2 diabetes, unless you're a Pima Indian, which is like this very, very high risk, which Dr. Leibel will talk to you about, group of people. So if you have a 5-year-old, they either have type 1 or some monogenic or secondary type. Like, you can't say it's definitely type 1 autoimmune, but you can definitely feel confident it's not going to be type 2. So the peaks are at 5 and at 13. And that probably has to do with how they grow, because as you are growing quickly, you need more energy. So your insulin demands go up. Just like pregnancy, gestational diabetes, your insulin demands went up. And so you uncovered a relative deficiency. So pubertal onset is going to be a thing that'll bring out diabetes, whether it's type 1 or type 2 or any type, because your demands go up. Interestingly, I didn't put it in here, but there were some studies done. There's a third peak of type 1 at age 60. And so when you see people and you see somebody who doesn't have any markers of insulin resistance, and they're 62 years old, and they have hypothyroidism and celiac disease, and no one in their family has ever developed diabetes, you have to scratch your head and think, that doesn't look like type 2 diabetes, where I would be seeing markers of insulin resistance and usually lots of family members. There's something unusual there, and they have other autoimmune disorders, so there is a peak in incidence around 60. Much more common in people of European ancestry because of genetic loading, risk for autoimmune diseases. And even though type 1 diabetes has an enormous genetic loading factor in terms of your risk of developing type 1, it's huge. Most people don't have anybody in their family who have it. So why would that be? So we'll talk a little more about it, but it's not entirely clear, but it seems that there's more than, it's not a single gene, it's not monogenic. It's related to their HLA type and your risk for autoimmunity in general. And if you think about organ donation and matching, or even when someone's going to donate their bone marrow, if you were trying to get a match, an identical match from your family member, that's not easy. And that's just HLA matching. It's not all the genes. So probably we think that there's just not a very high likelihood that everyone in, every one of those genes would be the same in another family member. So you're not going to have the same sort of identical risk. You have studies for kids who have type 1 diabetes who are identical twins. And that's very interesting because we believe there is an environmental factor. It's always how much is genetic, how much is environmental. So they looked at twins. We looked at them briefly, and we saw, wow, over 5 years, only 30% of the identical twins got diabetes. That makes it look a lot more like it's environmental. And then they kept watching and watching and watching, and we hit 30 years. By 30 years, 80% of the identical twins had it. So why one brother got it at 5 and the other identical twin brother didn't get it till 35, we don't know. But that's pretty strong genetic loading. It seems like if you live long enough, if you have those genes, the susceptibility is extremely important. But it's not unusual for people to say, this can't be because nobody in my side of the family has this. Everyone says that, but that's right. That's pretty much what we expect. We talked about that. So of the genetic risk, if we have both genetic and then environmental, of the genetic risk, the HLA types, that's only about half, 40 to 60%. So another 40 to 60% of the genetic risk is probably hundreds of other genes, and we're identifying them all the time that modify your risk. And as I said, there's been an increase in the incidence of type 1 diabetes in the past 20 years, and that's all over the world. It's not in one isolated place. It's everywhere, and it's not clear why that should be. Just like there's been an increase in type 2, honestly. But clearly, there's not enough time for your genes to have changed. The genetic composition of Finland did not change markedly in the past 20 years. So what is causing that? Some of it, obviously, is going to be environmental, and perhaps the environment's effect on the expression of genes. You're not going to change your genes, but you can easily change your gene expression. And Dr. Leibel will probably talk a lot more about that. So I understand you guys have already done immunology, so we're going to kind of skip over this too. But in terms of trying to understand why people get type 1, there's controversy over what is the primary antigen. And a lot of people believe it's insulin itself, that your body is rejecting your insulin. And not everyone, it's not clear that that's the case, but there's a lot of good evidence that that might be the case. And so if we could prevent that, then maybe we could prevent type 1 diabetes in people who are at risk. As I said, because nobody has family members for the most part, that means 95% of people with type 1, there's no one else in their family. How could we have found them, right? So, and only 5% of people who do have relatives with type 1 diabetes will ever develop it in their lifetime. So 95% of your relatives, if you have type 1, they're not going to get it. So how do we find the people who we could prevent it in? So that's a problem. But ideally, there's some defect in becoming tolerant to your own antigens, and ideally we'd be able to interrupt that early if we could identify how to change it. You guys already, we talked about this. Oh, the only other thing to know is that of the subtypes, DR3 and DR4 are your high-risk subtypes. If you have one, you're at a little bit of an increased risk. If you have both of them together, they're going to be more significant, even having DR3, DR4 together. And so if you do have that, you are considered the highest genetic risk. And so that's one of the groups of people we'll look at. So if you have a child, if you have type 1 and you have a child and they're DR3, DR4, their lifetime risk of developing type 1 is going to be 1 in 15. So that's really high. So that's a group we study. We collect their toenails, their growth markers. We take urine every year. There's something called the TEDdy trial. And there's a few other trials where you take people right from birth and you watch what they eat, when do they get their vaccines, everything, to see if we can understand why these very high-risk people, some of them will get diabetes and some of them won't. I put 40 to 50 here, but I didn't update that this year. There's hundreds of loci that we've identified now, and people with the decrease in cost and the increase in availability of whole genome scanning. We've been looking at our patients who look like they have something unusual. 3 kids, the father and the grandfather and the paternal uncle, they all have type 1 with autoimmune markers. You're like, well, it is autoimmune type 1, but that's really weird. There's something else going on in that family, and we can find things, and we have found things, and they're usually immunologic markers, some interleukin receptors, things like that. And then if you really probe that family history, everybody's got a lot of autoimmune disease. It's not just type 1. But on top of maybe being DR3 and DR4, having this gene really increased their risk, where everybody who had that particular gene, that inherited that gene along with their increased risk HLA types, they all got type 1. So it's interesting, and we're finding more and more out about that all the time. This is just showing over time how you see the incidence of type 1 diabetes increasing all over the world. Finland has one of the highest, next to Sardinia, prevalences of type 1 diabetes. And they're very homogeneous in their genetics. So, and they're small, and they're able to track their populations. So there's a lot of good information that can come out of those places. Finland in particular, they keep every piece of information in a general registry of everyone who lives there. So they can look back once someone gets type 1, and you can say, oh, what did they weigh when they were in the 3rd grade? And they can see. They've got it all. So it's a tremendous sort of database for information, but it is just all epidemiologic. But it's clearly not isolated to any particular area. We do see that people who have let's say, less developed countries do not have this degree of autoimmune disease. And so some people believe that that's significant because, I mean, it's certainly significant. Why is it significant? Some people believe it's because of the absence of antibiotics. You didn't change their microbiome. They don't get treated as much for infectious diseases. Some people believe, vaccines, they get less vaccinations in underdeveloped countries, and so you see less of it. I personally, while I do believe the microbiome has something to do with it, think there is probably something to what is also just a hypothesis called the hygiene hypothesis, which means that they get more antigen exposure. And we here have a lot less exposure with all our vaccinations. It's not the vaccines themselves, it's that you didn't get, those infectious diseases. And you didn't get worm infections in particular seem to hone your immunologic responses. So there's a lot of work looking at helminth infections and how they may hone your immune response, and why perhaps now that we have less and less of that in areas that have access to better sorts of water, clean water, and you just have less of those infections, you get more autoimmune disease, more allergy. And, it's a trade-off. I don't think you would want the level of infant and child mortality that comes along with not vaccinating your kids, right? That's still, but it may be why we see, more of it here than in places that don't have access to that. And if we can understand better, if that's really the case, then we can try to intervene and change that without having to just eliminate vaccinations and antibiotics. So this is something that is just supposed to reflect how we think maybe type 1 diabetes, but the truth is most of it is right for any type of diabetes, about how your insulin levels, and it says actually beta cell mass is reflected here over time, but it's a marker because we can't measure beta cell mass. Interestingly, there's very few good ways to measure beta cell mass. And there are people here studying it. How can we actually get in there and not just look at your pancreas, but see how much of their mass is islet and how much of the islet is beta cell? Because over time, when you have an inflammatory response in your islet, as you will in type 1 diabetes, you shift the content. So it used to be 70% insulin, so 70% beta cells, and now it's only 20% beta cell. The islet, it's still there, but it's not making, we think, as much insulin. Again, that's not a measure of how much it's releasing, so none of this is direct, it's all indirect. So when we're measuring beta cell mass, we're really just saying how much insulin can you produce. And when we're studying that in someone who's got to take exogenous insulin, we can't measure insulin, so instead we measure their C-peptide. So that's definitely a marker of how much of your total insulin you might be producing, how much is coming from your beta cells, C-peptide. So this beta cell mass is really being indirectly measured by your C-peptide production. And so what you get at birth, how much mass you have, that's going to be a potential issue as well. Maybe when you have more exposure to higher blood sugar levels in utero, lots more gestational diabetes, maybe that does something to your ability in terms of the amount of beta cells that you're producing, or maybe their function, maybe they're not normally functional. So certainly prenatally, there's a lot we don't know, and you could be born a little more challenged because of either your genetics or because of exposure in utero when you were developing the embryo. Then we think that for type 1, you have this sort of general period that could be anywhere from 6 months to 75 years, where you have pretty stable amount of C-peptide production. It's normal. It's what you were born with. It's fine. And then something happens. And you have this precipitating event, we believe, which, as I said, can happen when you're 5 or to your twin brother when he's 29. And that seems to trigger this autoimmune response. And a lot of people believe that most people, the antigen that's first attacked is insulin itself. And then your body is having this immunologic reaction, and you start to get inflammatory changes in the islet itself, where you have the beta cells declining in number. And then if you looked, which most of the time we don't know to look, you'd see a very gradual decline that will go up and down over time. We know now from the natural history studies of people that we follow over time. But still, even though you might not have normal insulin levels, you have normal blood sugar. And then eventually you get to a point where your C-peptide production is so low that you're going to be symptomatic. And somewhere in the 20% of what is expected, people will present with symptoms of type 1 diabetes. However, 20% is not functional at the time of diagnosis usually. Sometimes it looks like they make nothing. And the reason for that is that they don't function properly when they're exposed to all that hyperglycemia and abnormal lipid metabolism, and so beta cell function is affected. It sort of declines, and it's a sort of spiral. And then when you reverse that with exogenous insulin and you can restore normal lipid metabolism and reverse the insulin deficiency, and there's less exposure to hyperglycemia, you go into what they call the honeymoon period for type 1. The types of, what happens is the beta cells that are there, they start to become functional 4 to 6 weeks in, and then you can see what's left. Unfortunately, that's just still, we're in the decline. So even though that would be awesome if we could stop it right there, we can't seem to do it. We've done dozens and dozens of different immunologic agents. We just can't stop it. You can delay it by about 9 months. That's about it. You can change this curve, move it over by 9 months. So we're trying to figure out how we might be able to act earlier. Maybe if you could act back here, those agents would be more effective. We don't really know. Eventually, you'll go, continue to decrease in type 1. And as I said, actually in type 2 also. The biochemical autoantibody assays that I mentioned, we're discovering more and more of them, but the classical triad of biochemical antibody assays, meaning you just need serum and they measure them, would be insulin antibody, GAD65 or glutamic acid decarboxylase 65 antibody, or ICA512 or insulinoma-associated protein 2. So if you use those three in conjunction, it has the same sensitivity as using islet cell antibody, which is a tissue-based test and more expensive and difficult. So after looking at the fact that in relatively small studies, because as I said, this is an orphan disease, they decided they would say, oh, you can just do that triple screen. You don't need to do that more expensive tissue test anymore. But that might not have been really applicable to all populations, and we still do the actual ICA, which is the islet cell antibody with tissue. And there's a newer antibody called zinc T8. It's a transporter protein, and that's now clinically available as well. So we would measure these, and having any one of them is good enough to say you're having an autoimmune reaction. And as I said, GAD is very nonspecific. It's actually a neuronal protein, so it's not specific to type 1. But if you're looking at it in someone with type 1, we see GAD all the time in type 1 diabetes because of the innervation of the islets, presumably, and the inflammation there. I'm just going to go quickly at the next 2 slides, and then if you guys have any questions, we can answer them. This is a picture of what I was saying about the islets. Now, the golden brown part, that is staining positive for insulin, but all of this part is an islet. Ordinarily, this would be all the way out to the edges, and there would just be peripherally some glucagon staining. So while this cell, this islet clearly has some beta cells, they're very reduced in number, and there is inflammation there. Now, most of the time, most of our histopathologic information came from animals, because the pancreas is not easily accessed for biopsy at the time of diagnosis or later, right? So the only things we have now are people who are donating. There's a pancreatic donation group so that people who have passed away through traumatic accidents and things like that will allow us to harvest their pancreata and take a look at them histopathologically. And that's providing, the first information, honestly, over the past 10 years that we've had in terms of human histopathology of type 1 diabetes. And the rest of it has been inferred that it was similar to animals. So, sorry. So this is just showing you an islet with the staining for CD4 and CD8 positive T cells, not with any insulin staining, but there's a lot of infiltration there. And if you identify islet inflammation and islet autoantibodies, that doesn't mean that it got to the point where the person had diabetes. There are groups of people that we've studied and followed for over 10 years with 5 antibodies and decreased insulin levels. They are not normal, but they didn't progress to diabetes yet. Why? What's happening there? There's clearly some protective thing that they have genetics, something's protecting them from progressing the way we would expect. I'm not going to go into this, but this is progression in a NOD mouse, which, as I said, is what we've relied on for a long time. You just get, over time, the NOD mouse is a very accelerated model. They develop type 1 diabetes in a matter of weeks to months of life, so you can really see what's happening. And so over the course of 31 weeks, you can see that this mouse first had normal insulin, then had insulin, but look at all those inflammatory cells, and then had no insulin. And so that's what we think is happening also in general. I'm not going to go through this either. You know this. I think this got repeated. I apologize. I do want to just talk about type 1 DKA. So it is very important to recognize that there is no blood sugar level that is dangerous in and of itself in type 1 diabetes. In anybody, if you develop hyperosmolarity, right, that's dangerous. But that's not just related to a blood sugar level. There's a lot of other things happening there. If your sodium's 160 and your BUN and creatinine are high and you stopped urinating and your glucose is 1200, that's a huge, huge medical emergency. That's a big problem. But a kid who is type 1, who drinks a regular soda because he asked for diet and they gave him regular, he could have a blood sugar of 600. Not dangerous at all. It came fast, it went fast, he's done. What's dangerous in an insulin deficient patient is the basal insulin. That background insulin we talked about, that's what you need for your basic metabolic stability to stay alive, okay? So I could only take my background insulin, and I could have blood sugars of 600 all day. You know, that's going to increase my risk of retinopathy later on, but it's not dangerous acutely as long as I have the background insulin so that I can at least use some insulin, some glucose with the insulin for my background metabolic needs. When I don't have the basal, when I don't have my background insulin, I can't meet my basic metabolic needs. This is an emergency. That's when your body says, okay, it's an emergency. We're going to go to fat breakdown. We're going to make ketones as an emergency source of energy. The brain can use them if it needs to. It doesn't like to, but it can. That is a turning point. That's telling you you didn't forget to cover your hamburger. You know, on a roll, you forgot your background insulin. This is a state of emergency. It's a warning signal. So we teach people to check their ketones because that's, oh, I could have a blood sugar of 257, not that high, and large ketones and a bicarb of 2. That person could die versus someone who got a regular soda and has a blood sugar of 520 and, you know, doesn't have ketones. And that's unfortunate, but it's not dangerous. They don't need to do anything other than take the insulin to correct it. So background insulin deficiency is what's going to kill somebody. So you need enough to meet your basic metabolic needs. And ketones are a marker of that. You will never make ketones as a type 1 patient because you forgot to cover food. Teenagers come in and tell us that all the time. Oh, I forgot to take my insulin at dinner. No, you haven't taken any background insulin in days. Your pH is 6.9. You have not had any insulin in days. So that is going to keep them safe, okay? Not ideal control. Some of the development then from breaking down fat for energy into really going into diabetic ketoacidosis requires other counter-regulatory hormones. People who are deficient in glucagon, interestingly enough, people who've had their pancreas out for tumors, things like, they don't develop DKA. You need glucagon and some of those counter-regulatory hormones to develop DKA. Type 2 patients can if they become absolutely insulin deficient. But for the most part, DKA is much more common in people without insulin resistance because they're insulin deficient in an absolute sense. You're going to get a whole nother lecture about complications of type 1 diabetes, but I think most of you are aware of the common ones: retinopathy, nephropathy, neuropathy. Those are all microvascular diseases, okay? The retinopathy and nephropathy. You're talking about tiny little blood vessels versus cardiovascular disorders, stroke and heart attack, which are macrovascular effects. And then you'll get other things that are associated with it. If you are chronically hyperglycemic, you'll have increased risk of infection and poor healing. You can get connective tissue changes. So in the 90s, which wasn't that long ago, it was the first time that they had published a long-term study of why it was important to try to keep your blood glucose levels in type 1 diabetes in the close to normal range. They don't have to be perfect, but close, kind of, and that as you get closer, it actually did really decrease your risk factor for microvascular disease. And that was the first time it was proven. Now it seems like obvious, but back then that wasn't true. And what it also showed us was that as you approach 6, between 6 and 7, the relative risk came down very close to 1. So it's around 1.1. So you didn't need to get people with type 1 diabetes to have A1Cs in the non-diabetic range under 6, now under 6.5, but most people are under 6, to have them decrease their chances phenomenally of developing complications. It used to be everyone thought, oh, there was no question. Everyone with type 1 is going to have complications. Everyone's going to have a shortened lifespan. We do not believe that anymore. It's really hard work, but there is no reason to believe that people have to have complications, and their lifespan, if you're diagnosed today, we believe it will not be reduced. That is also what was found in cardiovascular disease with large vessel disease, that over time, when they looked back at the people that they had randomized in that diabetes control and complications trial to conventional versus intensive treatment, they found 42% risk reduction in the intensive management. So that was very impressive. This is just for you guys to have some idea of what has gone on here at the university that you attend and understand some of the historical importance. We were the second place in the United States that was able to give insulin to people in 1922. The first place was in Boston, the Joslin, and we were the second. Before it was available here in North America, in Canada, it was the very first place that a person was given insulin who had type 1. And this poor boy was diagnosed a couple of years before this was taken, when there was no insulin for anybody anywhere in the world. And then, in 1922, Columbia University was treating people. He lived here for over a year. And he was being treated with the only treatment of the time, which was starvation. Don't eat any sugar, don't eat any carbs, because you won't be able to metabolize them. So they would carb restrict them. And this is what happens when you carb restrict. So he was starving, and he was admitted, and he was started on insulin, and they measured a gazillion things, and we have that chart, like the actual chart, and we have the pictures and his caloric intake, and it's quite remarkable how much they were able to collect the information. And this was the same person right after, as he was being discharged, like a normal kid. He looked fabulous. That was just from letting him eat and take insulin, okay? So that was fantastic. And interestingly, his great-granddaughter was diagnosed with type 1 about 5 years ago. She did not even get admitted to the hospital. She came to the ER. We saw her blood sugar was high. Her bicarb was normal. She had some ketones, but small. She was very well appearing. She went home. She came back the next morning. She came back a few days in a row. And she was on insulin, and she's done great. She's on an insulin pump.